everyone. I'm Gigamon Punki. I'm Nafai Hayahama, and we are the member of project a comparative study of antibacterial efficacy of papaya seeds, Garcinia atroviridis fruit, and Rhodomyrtus tomentosa leaf.
Hello everyone, I'm Mr. Benjamin Cedeño. I'm an English as a foreign language teacher here at Naratiwa School, and I'm one of the mentor or advisor for this team's project.
Hello, my name is Arita Matavi. I am a teacher at Naratiwa School. I am also advisor for this science project. I am honored to participate in this program.
Hi, I'm Lalita Yeduson. I am a biology teacher in Naratiwa School. I was a mentor of this project.
Introduction. Staphylococcus aureus is a common bacterium that can cause a range of infections. In light of growing concern about antibiotic resistance, there is a strong interest in exploring natural alternatives. Traditionally, plant extracts have been used for their medicinal property. Our research aimed to investigate the antibacterial potential of extract from papaya seeds, Garcinia atroviridis fruit, and Rhodomyrtus tomentosa leaf against this bacterium.
The primary objective of our study were twofold. First, we aim to evaluate the antibacterial efficacy of extract derived from papaya seed, Garcinia atroviridis, and Rhodomyrtus tomentosa leaf. Second, we sought to compare the inhibitory effect of this extract, specifically against Staphylococcus aureus.
Based on our initial research, we formulated two hypotheses. Firstly, we hypothesized that the Garcinia atroviridis extract would inhibit the highest inhibitory efficacy. Secondly, we predicted that higher concentrations of the extract would demonstrate greater inhibitory effect on the bacteria.
Methodology. First, papaya seed extract preparation. Immature Garcinia papaya seeds were pulverized using a blender. A ratio of 40 grams of papaya seeds to 80 milliliters of distilled water was used. Blending for 3 minutes. The closed extract was centrifuged for 4,000 revolutions of 10 minutes. Two supernatant was collected using pipette and stored in amber bottle to prevent like exposure. Garcinia atroviridis and Rhodomyrtus tomentosa leaf extract preparation. Dry. Garcinia atroviridis and Rhodomyrtus tomentosa leaf virtually dry under sunlight for 3 days. 30 grams with 100 milliliters of distilled water for 3 minutes. The closed extract were let to sit overnight. The closed extract was centrifuged at 4,000 revolutions for 10 minutes. The supernatant was collected and stored in amber bottles at 1 to 5 degrees Celsius. Bacteria preparations. Staphylococcus aureus were obtained and inoculated into Newton bolt. The culture turbidity of adjust to 1.5 McFarland standard. The bacterial culture was then spread onto Miller-Hinton agar plate, 0.1 milliliters. Antibacterial activity assay. Extract from papaya seeds, Garcinia atroviridis, Rhodomyrtus tomentosa leaves, and distilled water were applied to paper dish, ensuring a 15 to 20 milliliters distance between dish and 15 milliliters from the plate end. The plates were incubated at 37 degrees Celsius for 20 hours. The zone inhibition were measured using a vernier caliper, and experiment was repeated 3 times. Part 2. Garcinia atroviridis extract preparation. Dry Garcinia atroviridis under sunlight for 3 days. 30 grams and 25 grams of Garcinia atroviridis were blended separately with 115 milliliters of ethanol for 3 minutes. The closed extract were let to sit overnight. The supernatant was collected and stored in amber bottle at 1 to 5 degrees Celsius. Rhodomyrtus tomentosa leaf extract preparation. Fresh Rhodomyrtus tomentosa leaf were blended with ethanol at ratios of 30 grams and 25 grams of leaf to 115 milliliters of ethanol. Blending for 3 minutes. The close extract were let to sit overnight. The supernatant was collected and stored in amber bottle at 1 to 5 degrees Celsius. Bacterial preparation. The bacterial preparation was the same as the part 1.
Finding. In part 1, here we see the result of our experiment with aqueous extract as shown in table 1 and figure 1. The Garcinia atroviridis extract exhibited the highest average inhibition zone of 12 mm. Papaya seed and Rhodomyrtus tomentosa leaf extract show minimal inhibition zone with 6 mm each. Therefore, the Garcinia atroviridis demonstrated the most significant antibacterial effect using water as a solvent. In part 2, using ethanol extract, we observe even more pronounced effect. Table 2 and Figure 2 show that the Garcinia atroviridis extract at a 1:2 ratio yielded the highest inhibition zone of 22 mm. Rhodomyrtus tomentosa leaf also show inhibition, but Garcinia atroviridis was considerably more effective. Therefore, the Garcinia atroviridis extract with ethanol at a ratio of 1:2 yielded gave the best result.
Discussion. The study of Staphylococcus aureus inhibition using extract from papaya seeds, Garcinia atroviridis, and Rhodomyrtus tomentosa leaf. The Garcinia atroviridis extract demonstrates superior inhibition of Staphylococcus aureus compared to the papaya and Rhodomyrtus tomentosa leaf extracts. The Garcinia atroviridis extract inhibition of Staphylococcus aureus alix, with the finding of Panuvat Kongno, who conducted experiment on the inhibition of Propionibacterium agnes, where Garcinia atroviridis extract inhibitory effects. In this project, the Garcinia atroviridis extract was also used in inhibit Staphylococcus aureus, and it proved to be the most effective compared to the papaya and Rhodomyrtus tomentosa extract. The Garcinia atroviridis extracts yield an average inhibition zone of 22 mm against Staphylococcus aureus.
Based on our result, we recommend several directions for future research. Firstly, testing higher concentration of the extract could further enhance their inhibitory effect. Secondly, identifying the specific active compound would be valuable. Finally, exploring the potential of incorporating Garcinia atroviridis extract into product design to combat Staphylococcus aureus infection is promising. Future tests on other bacteria could also be beneficial.